Jennifer Doudna, Ph.D., Nobel Prize in Chemistry. In the biography of Doudna, found on Proceedings of the National Academy of Sciences, she is said to have first become interested in science, taking chemistry in high school, participating in a science seminar series highlighting the chemistry of biological systems. Furthermore, upon reading The Double Helix on the discovery of the structure of DNA, Doudna was hooked and desired to study deeper into the mysteries of life science. She studied chemistry at Pomona College, Claremont, California, where she was impacted greatly by several professors on her research career and line of study, beginning her first scientific research here, working in the laboratory of Sharon Panasenko, her undergrad advisor. Panasenko became a strong female role model for Doudna and provided her with guidance early on in her scientific career. After her undergrad, she attended Harvard Medical School and earned a Ph.D. in biological chemistry and molecular pharmacology. She mentions growing up having to overcome bullying in the process of knowing who she is and how it has become a part of her ever since. An interview blog with the National Institute of General Medicine Sciences reveals that one of Doudna's most prominent hobbies is gardening. If she's not in the lab, you're most likely to find her amidst the Berkeley garden fruits and vegetables. She finds it an intellectual challenge to coax plants from her childhood home to grow in Northern California as well. Loving to get her hands dirty and challenge herself in other ways, gardening allows her to reconnect with nature and relax. I admire Jennifer Doudna for providing an example of someone who persevered through all the hard work, pushing the boundaries of science and making groundbreaking discoveries. Growing up, I was surrounded by science in the same way that she was. My mother was a biology teacher at my high school, and being a part of her classes and growing up seeing the passion for the study is part of why I'm majoring in it right now. Doudna's persistence, being a well-respected female scientist who has made great strides in not just the science community, but medical as well, gives me the sense that I too can accomplish similar feats. Her work with molecular biology is astounding and something that I would love to study and be a part of someday too. In an interview with Doudna, the Nobel Prize website, she tells readers to embrace your interests and passions and just go for it. She says to keep pursuing what it is you love, and I think that alone creates a deep sense of admiration for her and her work. Jennifer Doudna, most well-known and foundational research is on CRISPR-Cas9 genome editing. Her lab's research led to the discovery of CRISPR, a tool that is used to make targeted changes to the genome. Found in bacteria cells, CRISPR is used in immunity. It takes part of the virus's genome and inserts it into the bacteria's genome, creating a guide RNA sequence so that the next time the virus infects the cell, guide RNA recognizes it quickly and is able to bind and destroy it. While this is the most profound of her studies, she has several other research expertise in RNA machines, hepatitis C virus, RNA interference, ribosomes, X-ray crystallography, and cryo-electron microscopy, says UC Berkeley Research. A recent scientific article that Jennifer Doudna was an author of was on the discovery of ancestral CRISPR-Cas13 ribonucleases. According to the article, prior to the study, it was known that DNA-targeting class II CRISPR-Cas enzymes, including Cas9 and Cas12, likely emerged from transposon-encoded nucleases. However, the origins of RNA-targeting Cas13 systems have been unknown. In the figure below, depicts the investigation of Cas13AN protein for biochemical analysis due to its vitro stability. Gel electrophoresis experiments displayed in A and B revealed that mutations in the conserved RX4H motifs of the heparin domain inhibited cleavages, and the presence of Mg2+ cleator EDTA also inhibited Cas13AN activity, showing that unlike its larger counterparts, Cas13AN uses a single active site for both CRISPR RNA processing and RNA-guided cleavage, revealing that ancestral nuclease domain has two modes of activity. Jennifer, as mentioned previously, has already uncovered the genome editing tool CRISPR. She has since expanded studies further uncovering the range abilities of CRISPR-Cas9. It has since been used in numerous clinical trials by medical professionals for treating disease, diagnosing disease, and even engineering agriculture. According to UC Berkeley Research, Doudna and her team are currently focused on discovering and determining the mechanisms of CRISPR-Cas and associated proteins, developing genome editing tools for use in vitro, plants, mammals, and developing anti-CRISPR agents. Science would not be where it is today without the discoveries of Jennifer Doudna. Her profound studies and dedication to research and education over the years has impacted the science community as well as our progress in medicine. She's become a significant role model for females in STEM, providing an example of overcoming boundaries and pushing past what is known. Her discovery of CRISPR will continue to be developed and refined as time goes on, and her work will be set in stone for others to be able to learn and build upon.